Friends, today's topic is schistosoma life history. Yesterday we discussed about the schistosoma structure, okay, and the schistosoma haematobium, female ecosystem and male ecosystems is over. And today we are discussing about the life history, schistosoma haematobium life history. Here, schistosoma haematobium, it is one of the Diazinian parasite. Diazinian parasite, which means they need two hosts. Primary host is the man and secondary host is the freshwater snail. Freshwater snail, మంచినీటి నత్త అని చెప్పుకుంటాం కదా, అది ఇంటర్మీడియట్ హోస్ట్గా లేదా మాధ్యమిక అతిథి లేదా సెకండరీ హోస్ట్గా ఉండడం జరుగుతుంది. సో హియర్ schistosoma is a male and female. These two are the male and female are always attached with the help of the gynecoporicle having the male. So here the spermatozoa is released from the male of the male schistosoma, and the ovum also released from the female of the schistosoma. These both are contact together and form a zygote. So the process is called as a fertilization. Here it is a gonopore. This is the gonopore of the female. In the from the male, the spermatozoa is come from the male schistosoma, and they have to tail. That's why they can move. They can move from the male of the body of the male to through gonopore of the female. So the spermatozoa is come from the male from the schistosoma, and which are these two are the contact together in the oviduct. Here, so they can move, and the already ova is released from the ovary, and which are traveling and which are stable in the oviduct. The spermatozoa having the tail, that's why they can move from the to respective way. That means gonopore and ootype, uterus and ootype and the oviduct. So these in the oviduct of the female, here spermatozoa and as well as ovum, these two are the contact together and form as a zygote. That is the zygote is a two stage of nucleus. Because sperm also have the end stage of nucleus and the end stage of chromosomes, and they also end stage. These are the zygote is the two stage. So the fertilization process occurs in the which place? Oviduct of the female. Okay, when the completion of the fertilization process, here one vitelline glands are available. Vitelline glands are available in the female of the body, of the schistosoma. They are releases some secretions. And the same thing, the vitelline glands are having one type of secretions in there, and they are producing yolk cells also. These yolk cells are here. This is the nucleus, and these are the yolk cells. This is the yolk cells. Yolk cells is nothing but it is used for the feeding. This is the nucleus. This is called as a zygote. When the vitelline glands are releases as produced some secretion surround the zygote. This is shell. The cells never form చేస్తున్నారంటే vitelline glands నుంచి వచ్చిన secretions cells ని form చేస్తున్నాయి. So this is the nucleus, this is the yolk cells, this is the vitelline gland secretions. That means cells. So the complete structure is called as a capsule. Around 10 to 100 nucleus or capsules are available in the uterus of the female. So nearly 100 నుంచి 10 to 100 capsules ఇందులో మనము చూస్తాము ఎక్కడ యూటిరస్లో ఉండడం అనేది చూస్తాం. ఇది ఇక్కడ 100 నియర్లీ 10 to 100 యూటిరస్ క్యాప్సూల్స్ అనేటువంటి ఇందులో ఉండడం అనేది చూస్తాం. సో this is the zygote. Now come to here structure. This is the oral sucker. This is the male and this is the female. Okay, which is in the blood vessels. Which prevails the blood vessels lo undadam అనేది జరుగుతుంది. This is the adult and the here zygote is formed, that is the nucleus and cells. And this is the yolk cells and the nucleus. This is the capsules. This is the capsule. Here, so the adult worms are live in the blood vessels of man. So when the fertilization process is occurred, the capsules are formed. All the capsules are traveled from the blood vessels to urinary bladder of the man. So the capsules are is divided with the along with the urine. When the urine is diluted with the water, the capsule will be completely encapsulated. That's why some of the one larva is released from the egg. So that is the first one larva is the first larva is called as a Miracerian larva. This is the Miracerian larva. So the body is covered with cilia. This is the cilia. The body is covered with the cilia, having two penetration glands also there. In anterior part is there are two penetration glands is there, and the middle of the body having the two flame cells also there. Flame cells, and this is the cilia, and this is the penetration glands. Okay. Having with cilia, they have to move, which is only live in the 24 hours only, which is waiting for the Miracerian larva is, this is called as a Miracerium. This is the first larva of the Schistosoma. Okay, this is the Miracerian larva, and which is only lives in the 24 hours. Within the 24 hours, they can found in the freshwater snail, they can enter to the snail. If it is not found in the freshwater snail, they can die. 24 hours లోపు అలా దొరకకపోతే మాత్రం ఏమవుతుంది, అది చనిపోవడం జరుగుతుంది. సో విచ్ ఈజ్ వెయిటింగ్ టు ఇంటర్మీడియట్ host, that's why when the if it's possible freshwater snail is come from that area, the Miracerian larva is entered to the snail. Okay, the penetration glands are helps to the penetrate the skin of the snail, and when they enter into the Miracerian larva, the cilia will be completely lost. When they enter to the snail, the cilia of the Miracerium is lost, and when lost the cilia, they can enter to the snail. This directly enter to the visceral and some of the parts, and here this is the sporocyst and the merocyst in the snail. So Miracerian larva is a infective stage 2. What is that? Infective stage 2 snail, freshwater snail. Okay, and this is the sporocyst. So Miracidian larva elongates and develop and changes as a tubular structure called as a sporocyst. The sporocyst larva is developed in the freshwater only, freshwater snail only. Okay, so here this is the sporocyst, and the sporocyst contains the secondary having the germ cells. This is the germ cells. This is the primary sporocyst and the secondary secondary sporocyst. Secondary sporocyst here. So Miracidian larva enter to the with the help of the penetration glands. The skin is cut down the snail and the enter to the snail, and the which is Miracidian larva is entered to the snail. The cilia of the Miracidian larva is completely lost, and when they enter to the snail, the oval shape of the Miracidian larva is changes as a tubular structure that is called as the sporocyst. Okay, sporocyst. The sporocyst also tubular structure, and it is the second larva of the schistosoma, and it is a having the germ cells and having the germ cells, and it is also changes as a secondary sporocyst. In this, after the first primary first sporocyst and secondary sporocyst is over, the Redia larva is not occurs in the schistosoma. సిస్టోజోమాలో మనకి ఇటువంటి రీడియల్ లార్వా అనేటువంటిది మనకు కనిపించదు. ఎందుకు అంటే ఓన్లీ మిరాసిడియన్ లార్వా, స్పోరోసిస్ట్ లార్వా, సర్కేరియా. యాక్చువల్లీ ఫ్యాషన్ ప్లాటినస్ ఏమైనా ఉంటుంది, మిరాసిడియం అండ్ ద స్పోరోసిస్ట్ అండ్ రీడియా అండ్ సర్కేరియా అండ్ ద మెటాసర్కేరియా. దిస్ ఈస్ ద level of the, this is the sequence of the larva stages, but here only we have three Miracidium and the sporocyst and Cercaria. Only these three larvae are found in the Schistosoma life cycle. So after the, so this is the life cycle. This is the life cycle. This is the life cycle in fish, in snail only. ఇది మన నత్తలో చూస్తున్నటువంటి ఏంటి వాట్ వి కాల్ లైఫ్ సైకిల్ ఇన్ ద స్నేల్, నత్తలో ఉన్నటువంటి జీవిత చరిత్ర మనం చూస్తున్నాం. ఆఫ్టర్ ది స్పోరోసిస్ట్ దే హ్యావ్ ద gonopores is there. The sporocyst having the gonopores there. In this gonopore, some of the Cercaria larvae, in the secondary sporocyst having the in the posterior portion have the some of the Cercaria larvae also there. Cercaria larva. So tell me, Cercaria Cercaria larva is also developed from the sporocyst. Same thing. Miracidian larva is come from the hatch of the egg, and the sporocyst larva is the from the Miracidian larva, and the Cercaria larva is from the sporocyst larva. So here this is the Cercaria larva. Here this is the cystosome, this is the oral sucker, and this is the oral sucker, and this is the ventral sucker, and this is the fork. Agada. So this is the Cercaria larva. Cercaria larva having the two suckers, oral sucker and the ventral sucker, and the in the tail is there, that is the fork type of tail. The nearly body length is the 200 mm length. The fork tail is the 75 mm of the length of the body. So the length of the body is the 200 mm and the tail is the only 75 mm of the length. And which is Cercaria larva is entered to the man's skin. So to contaminated water or contaminated food, uh, when the Cercaria larva is directly entered to the man's. So if it is the man, if the man is taking for the bathing or washing the legs and hands, when the Cercaria larva having this the some of the glands is available in the posterior part, anterior part of the Cercaria, which is helps to the cut down the skin of the host, that is the man's host. Man is the host, first primary host. So Cercaria larva is the infective state to the man, and the miracidian larva is the infective state to the snail. So when the Cercaria, when the person is taking from the water contaminate, water is already contains the Cercaria's larvas. The directly they penetrate the man's skin and cut down the skin and enter to the man's skin. When they enter to the man's skin, there will be blood vessels as well as heart and lungs and the hepatic region. అక్కడికి వెళ్తుంది అంటే ఆఫ్టర్ దట్ pelvic vein వెళ్తుంది అంటే the man's enter to the skin through blood vessels after the heart. They can move from the one place to another place. So man's skin to blood vessels to heart to lungs to hepatic region to pelvic vein. In the last year, pelvic vein here in this one, they are the matured. Pelvic vein matured. They can, after the matured, they can also during the sexual life. After the sexual life, they can also release some of the capsules. This is the Schistosoma haematobium life cycle. Okay, I'm telling once again, just a summary. This is the adult worms. This is the adult worm of the female. The gynecoporic canal is there. The female is always attached to the male along with the with the help of the gynecoporic canal. So these two are the living in the blood vessels of the man. So and spermatozoa is from the male and ovum is from the female. These two are the contact together in the oviduct of the female and the form as a capsules. Actually, end stage of spermatozoa and end stage of ovum. These two are the contacted and form as a 2-year stage of zygote. This is the nucleus. Okay, and the vitelline glands of the female have releases some of the secretions as well as some of the produced some cells, that is the yolk cells. The yolk cells are using for the feeding, and this is the zygote and this is the yolk cells. The vitelline glands secretes secretions that is surrounded to the zygote and yolk cell, that is called as a shell. The complete structure of the shell and nucleus and the yolk cells is called as a capsule. Nearly 10 to 100 capsules are available in the uterus of the female. When the capsules are traveling from the blood vessels to urinary bladder of the man's. So the man's is releases the urine. The capsules also releases along with the urine. When the capsules are attached to the or diluted with the water, the capsules is encapsulated. క్యాప్సులున్నటువంటి కోశం ఏదో సిస్టమ్ ఏదో అదంతా కరిగిపోయి క్యాప్సుల్స్ నుంచి ద ఫస్ట్ లార్వా ఈజ్ ఎంటర్, దట్ ఈస్ ద miracidian larva. The miracidian larva is a oval shaped. Here the body is completely covered with the cilia. That's why they can move. అంటే మూమెంట్ చూపించడం అనేది జరుగుతుంది. And one pair of penetration glands is formed in the anterior part of the miracidian larva, and one pair of flame cells is there. The flame cells are used for the excretion, and so which is waiting for the intermediate host of the intermediate for the, that is the snail, which is live only 24 hours. After the 24 hours, which is die. If found in the snail, it will be entered to the snail, but it is not found in the snail, it will be die. Okay, and that's why it is the infectious stage of the snail. So in the this miracidal larva have a posterior portion of the some of the posterior some of the cells is there, that is called as a germ cell. The same germ cells are produce the sporocyst. So when the miracidal larva enter to the snail body, and we enter to the miracidal larva, each changes has a tubular structure, that is the sporocyst stage, and the sporocyst stage also having the posterior portion germ cells also there. Okay, and the sporocyst larva is lives in the liver in the snail. నత్తలో ఉన్నటువంటి లివర్ నందు అది లివ్ చేస్తుంది. And that is a primary sporocyst, and this is the secondary sporocyst. This secondary sporocyst also having the same cells, same structure, tubular structure, and having the cercaria. The sporocyst larva have the gonopore. The cercaria larva is expelled from the gonopore. So అంటే ఇక్కడ ఏమవుతుంది, స్నేల్ నుంచి బయటికి వచ్చేస్తుంది. అంటే స్టేజ్లో కనిపించేటువంటి ఒకటి ఒక దశ ఏంటిది, మిరాసిడల్ లార్వా స్పోరోసిస్ దశ మాత్రమే స్నేల్లో కనిపిస్తుంది, మిగిలిన దశలన్నీ కూడా బయటకు వచ్చేసి ఉంటుంది. సో సర్కేరియల్ లార్వా ఈస్ ఎక్స్పెల్డ్ ఫ్రమ్ ది స్నేల్ బాడీ which live in the water, lives in the water. They have to, there are the two suckers is there, oral sucker and ventral sucker, and having with the forked tail is there. The length of the circular larva is the 200 mm of the length, and the forked tail is the only 75 mm of the length. This is the circular. The circular larva also lives only 1 to 2 days only, which is waiting for the primary host. So they can live only 1 to 2 hours, means 24 hours to 48 hours only matrame. But in that period they can found the primary host, they can enter to the man's skin. But it is not found in the primary host, they can after 24 hours, after 48 hours they can die. So the mode of the infection is the contaminated water, or swallow the water, or the washing with the water, or the bathing with the water. The circular larva enter to the man's. Okay, so man's skin is cut down the with the help of the penetration glands of the circular larva. So once enter to the man's, the skin is captured and enter to the blood vessels, and they can move from the blood vessels to heart to lungs to hepatic region, and hepatic region is enter to the pelvic vein, and the pelvic vein, when they reaches to pelvic vein, they can mature in the pelvic vein, and they are ready to sexual life, and again they enter to the blood vessels. They can start with the life cycle, and they can also ready to the sexual life, and they form as a zygote, and that is the capsules. This is the Schistosoma haematobium life cycle, and tomorrow we are discussing about another topic. Okay, thank you.